Hello and welcome to the presentation where we shall discuss association networks in microbiome, some of the current outstanding and often overlooked issues, and their novel solutions. My name is Riabchenko and I'm a PhD student working in Firstland's lab of Max Delbrück Center in Berlin. First of all, association network analysis is a popular method for exploratory data analysis for identifying relationships between members of a microbial community, the wider ecosystem, and other relevant data, such as clinical data, in a statistical analysis pipeline. It's usually visualized as a graph or a matrix that contains relevant association metric of choice between the objects. Microbiome data poses many challenges, and many methods have been developed so far to solve them, but they often have single issue solving mindset, often failing to address multiple problems systematically and at once. Some of those usual problems include compositionality of the data, confounders and heterogeneity, complex and nonlinear interactions, zero inflation over dispersion, high dimensionality of the data, and benchmarking difficulties. All of those issues have methods to solve them, but they are usually not considered as a part of a singular statistical pipeline. Some of the most overlooked issues in microbial network research would include ignoring the impact of compositionality of statistical performance of the tool set, unreliability of certain tools because they self-evaluate themselves on the same model that they use, lack of additional information and variables such as directionality and causality, lack of comprehensive tools for nonlinear and longitudinal data analysis. So for example, we see here a lot of novel tools fail completely when posed with nonlinear relationships within the data. And as there is an example of very nonlinear data relationships both at a singular point of time and over time. Analysis of high-order interactions and deciphering ecological context of those interactions is usually lacking. And also, simulated data does not match real data well. So for example, in this principal coordinate analysis of real data and simulated data done by different methods, we can see that the simulated data fails to reproduce the two dense clusters of the real data, being more uniformly dispersed over the dimensions. Many solutions have been proposed to many of those problems, but first and foremost, we should note that the most effective way to approach data analysis of microbiome would not be to use those methods one by one, but to create a pipeline that integrates interdomain data, confounders, and different methods, and also accounts for all the data characteristics such as compositionality and zero inflation. One of the examples of such a pipeline that has been used during several projects in the lab would be displayed here. So basically, we account for compositionality, for the zeros, for the missing data, analyze the data to come up with the best normalization method and feature selection to trim the data, and then choose appropriate models to create association metrics and then visualize it. There are many ways how environmental and interdomain data can confound associations within microbiomes, but it can be accounted for with different approaches, such as creating homogeneous subsets of data based on environmental data, filtering the data after calculating association metrics, using environmental data as nodes within the networks, and deconfounding, so to speak, the microbiome data based on regression models. To treat compositionality, family of log transforms is often used, but it has a zero problem, which can be amended by adaptive pseudo-counts method or zero classifying strategies to reduce the bias. Alternatively, hierarchical models can be applied that account for compositionality of the data. New approaches with nonlinear metrics like distance correlations and Hilbert-Schmidt independence criterion for network construction can be used to account for data with complex relationships. Simulation tools like Simba or versions of normal to anything for complex settings and realistic results can be used. New approaches for network constructions for longitudinal data, such as compositional generalized Lotka-Volterra equations, can be used to account for both the time dimension and the compositionality of the data. Also, neural networks and Markov chain Monte Carlo methods are widely used in analysis of interactions in genomics data. Naturally, it follows that we could potentially use those methods for microbial associations. For the inference of the network itself and the direct associations within itself, a lot of efficient algorithms have been developed, like height-on-PC. Those methods not only infer direct relationships, but also can assist in predicting edge directionality. Those methods can be roughly divided into time series networks, correlation methods, regularized models such as speakeasy, probabilistic algorithms and hybrids such as height-on-PC, and information-based methods such as tools that rely on mutual information. An example of a network that was created by the above-mentioned pipeline and a height-on-PC based tool FlashWeave is this sparse network of direct associations for CLD clinical parameters and microbial, viral, and functional and fungal data. Finally, the references for this presentation. I would personally recommend everyone to read Statistical Dependence Beyond Pearson's Law by Jostheim et al. Thank you and goodbye.